for the defense, I'd like again to thank my supervisors and examination committee for their time and effort they took in evaluating my thesis and also in guiding my PhD project. So as Annette already mentioned, my topic was on, or my title was the assessment of genome-wide somatic copy number alterations in combination with driver mutation analysis and circulating DNA of colorectal cancer patients. Within this presentation, I'd first like to give you some background on the topic itself, then tell you something about the rationale and methods for this project. And then I have two larger chapters or parts of the talk. One is the targeted analysis of circulating tumor DNA, which was kind of the first project of my PhD. And the second part would be the untargeted analysis of circulating tumor DNA, which was also the second part of my PhD. And finally, I would like to conclude with the contribution of this PhD project for future medical care. But now, starting with the background. Colorectal cancer develops through defined stages from dysplasia over adenomas or serrated polyps to colon carcinoma. And there were or are two major pathways described. One is the adenoma carcinoma pathway that is initiated by a variant in APC, followed by variants in NRAS and KRAS, and followed eventually by variants in SMAD4 and TP53. And colorectal cancers that develop through the adenoma carcinoma pathway are eventually leading to microsatellite stable colorectal cancers that are associated with high numbers of somatic copy number alterations. And the second most frequent pathway is the serrated neoplasia pathway that is initiated by a variant in ctNNB1, followed by variants in KRAS or BRAF, and finally followed by PIK3CA and TGFBR2 variants. And colorectal cancers that develop through the serrated neoplasia pathway are eventually leading to microsatellite stable or microsatellite unstable colorectal cancers and are associated with lower numbers of somatic copy number alterations. Once a colorectal cancer is diagnosed, there are distinct molecular biomarkers that are used to guide treatment and to perform prognosis or get prognosis or predictive information about the course of disease and the patient's outcome. And one biomarker that is frequently analyzed is CEA, or carcinoembryonic antigen, that is already analyzed at baseline to predict whether the patient is most likely to suffer early recurrence, and then at multiple time points throughout treatment to assess the response to treatment, and also in follow-up examinations to predict whether there might be disease recurrence. Then the probably best or most validated prognostic biomarkers are MMR, MSI status, and DPD. And those markers are analyzed following surgery to assess whether the patient should be treated with adjuvant chemotherapy or not. And only in metastatic colorectal cancer patients, there is also the molecular status of RAS and BRAF, so whether there are genetic variants present in RAS genes or BRAF, and those information are used to refine the treatment strategy. So, for example, to enable some kind of personalized treatment. In general, specifically, those genetic variants are analyzed from tissue biopsies. However, tissue biopsies are invasive and do not accurately represent the entire tumor heterogeneity, as they are taken from one distinct tumor site. An alternative where lots of research and also, lots more is going on within the recent years are liquid biopsies. And liquid biopsies enable the analysis of circulating tumor markers. They might enable serial monitoring, as simple blood withdrawals are required rather than invasive biopsies. And they might represent the entire tumor heterogeneity in a more detailed way, as circulating tumor markers are shedded from all tumor sites, so also metastasis and not only the primary tumor. Within my project, I focused on the analysis of circulating tumor DNA, as this is the circulating tumor marker that is probably the easiest accessible and also most promising to use already in a clinical diagnostic setting. And circulating tumor DNA represents a fraction of the entire cell-free DNA and is detected based on cancer-specific alterations. And the probably best well-established way to detect circulating tumor DNA are targeted ctDNA analysis that focus on distinct hotspot variants that are known to reoccur in cancer or solid tumors. However, not all tumors present with such hotspot variants, and therefore an alternative approach such as untargeted ctDNA analysis are actually really promising, as in this case, you are not dependent on the presence of hotspot variants to detect ctDNA. And those untargeted analysis can focus or detect chromosomal rearrangements, copy number alterations, DNA methylation, chromatin signatures, and global or regional fragmentation. And those features specific for ctDNA are present in almost all tumors. So far for the background. Now, what did I actually do within my PhD project, or what was the plan? So as I just mentioned, there are distinct cancer-specific alterations that can be targeted to identify circulating tumor DNA. And within my project, I developed two methods. One would be droplet digital PCR. And here I validated two or more assays, but specifically two assays that are associated with colorectal cancer. One would be targeting KRAS PG12 PG13 variants, and the other BRAF PV600E variants. So distinct hotspot variants that are known to be present and frequently present in colorectal cancer patients. And then I also developed a second method that is whole genome sequencing based, and we called it live CNA. I will tell you later more about this, but this approach specifically identifies copy number alterations, chromatin signatures, and global and regional fragmentation. So yes, I was working on two different methods, and I wanted to assess whether those approaches might be used for residual disease detection, treatment monitoring, or even the early detection of colorectal cancer. And overall, or to assess those clinical questions, a study cohort of 152 participants was recruited, including 66 CSC patients and 86 healthy controls. And of those 66 CSC patients, 22 were analyzed with ddPCR, either for the detection of molecular residual disease or for treatment monitoring. And 50 of those patients were analyzed with live CNA, so the untargeted approach I mentioned earlier, and also either for the detection of residual disease or for treatment monitoring. Within the next part of this talk, I'd like to focus on the targeted analysis of circulating tumor DNA based on droplet digital PCR. And droplet digital PCR enables the distinct detection of specific hotspot variants by partitioning cell-free DNA following isolation into up to 20,000 droplets that contain then either wild-type alleles only, circulating tumor DNA, so mutant alleles only, or both alleles. And by performing specific PCR for the target of interest and using Tagman probes that are labeled with a fluorescent dye based on whether they are detecting or binding to wild type or to the variant of interest, it is then possible with droplet digital PCR, so by analyzing the fluorescent signal in each distinct droplet, to obtain information about how many droplets contain wild-type template only, how many contain mutant template only, and how many droplets contain both templates. And with this analysis, it is possible to identify the allele frequency of the variant that is specifically associated with circulating tumor DNA. But there are distinct analytical requirements that need to be met to use liquid biopsy in clinical practice. And therefore, within this project, I established or validated distinct cutoffs, either for the detection of lowest levels of ctDNA, so the limit of blank, and that is critical for once when I want to make treatment decisions based on the presence of hotspot variants, that might provide useful information for targeted treatment, or also for the detection of residual disease post-surgery or post-treatment to identify whether circulating tumor DNA is still present and might indicate the presence of remaining tumor cells. And then a second critical cutoff for the analysis of circulating tumor DNA in clinical practice would be the limit of quantification, because only above this cutoff it is possible to reliably quantify circulating tumor DNA levels and predict, for example, response or resistance to treatment. And within the first paper of my PhD, I've focused specifically on the analytical validation of 5 ddPCR assays. The rationale behind it, or actually the idea, was that most clinical assays so far, or assay providers also, only provide information about the sensitivity of the assay, so the limit of detection, which is the cutoff from which reliable detection of the variant is actually possible. But this cutoff doesn't really help for clinical interpretation, as actually it would be critical to know the cutoff, the limit of blank that represents the cutoff for 95% specificity, as from this cutoff, actually true positive signals can be reliably distinguished from the background noise. And then if you want to use liquid biopsy assays for treatment monitoring, the limit of quantification is also critical, as this represents or is based on the assay-specific dispersion, and only above this cutoff it is possible to reliably quantify the variant allele frequency. So as mentioned in the slide before, therefore the limit of blank is the critical cutoff for ctDNA positivity and negativity that might be used for residual disease detection, and the limit of quantification is the critical cutoff for treatment monitoring, as this is critical to reliably quantify ctDNA. When looking at the 5 assays that were validated within this study, you can see here each of those panels is one cutoff, so limit of blank, limit of detection, and limit of quantification, and each row represents the results obtained for one assay. And the x-axis actually represents the variant allele frequency that is associated with, or that is the cutoff specifically. And what you can see when you look at those 5 assays for the limit of, or the limit of blank of those 5 assays, you can see that they are widely dispersed. So it's not possible to validate only one assay for one specific variant and use this cutoff then for all the future assays you want to use. But it's actually important in a clinical setting to know the distinct cutoff from which it is possible to reliably detect ctDNA. And similar results were also obtained for the limit of quantification, as you can see here. Depending on the assay, the performance or the cutoff for reliable ctDNA quantification is also widely dispersed. Within the second paper of my PhD project, I wanted to also clinically confirm that those cutoffs are useful for, sorry, my phone is ringing, that those cutoffs are really useful for clinical interpretation, and therefore 42 study participants were included, including those 22 CSC patients I previously mentioned and 20 healthy controls. And the patients were either analyzed for residual disease detection or treatment monitoring. And when looking at the patients, the 18 patients that were included for the analysis for residual disease detection, here at baseline, it was possible to detect ctDNA more reliable, or in more patients with increasing disease stage, above the limit of blank. So in stage 1, it was unfortunately not possible to detect ctDNA at baseline. And following surgery, only in 3 patients, ctDNA was still detectable, indicating residual disease. In 2 of those patients, those were stage 4 patients, they still had metastases that weren't removed during surgery. So it was known that there were remaining tumor burden. And this one patient actually was graded already or classified as high-risk patient, and this patient also developed disease recurrence more than 1 year after surgery. for disease monitoring, so this is just an example of one patient that was followed throughout the course of disease. And as you can see in this upper plot are actually the results obtained from ddPCR analysis. You can see here that at diagnosis, it was not possible to detect circulating tumor DNA, also therefore not following surgery, but this patient developed a disease recurrence, and kind of in parallel to disease recurrence, it was possible to detect ctDNA again that was not present following surgery anymore. But here, more than 3 months before the second disease recurrence, as you can see here, obtained or measured again ctDNA signals indicating disease recurrence in this patient quite some time before it was clinically confirmed or clinically found. And then following the initiation of chemotherapy, ctDNA levels dropped again. And in parallel to the finding of stable disease, ctDNA was not detectable anymore. And just as a reference marker, CEA, the frequently used biomarker for CRC patients, was analyzed. But all this, what could be observed with ctDNA analysis, could not be seen based on CEA analysis, as measurement results were always below the threshold, so what weren't indicating the presence of cancer. Yes. So in summary, ddPCR enables the reliable detection of hotspot variants, and the limit of quantification, limit of blank could actually established to be a reliable cutoff for ctDNA positive signals, and also that can be used for residual disease detection. And the limit of quantification could be verified as reliable cutoff for treatment monitoring. And overall, for CRC patients, the ddPCR could be shown to be a highly sensitive and highly specific approach to detect specific hotspot variants. But in total, only around 44% of patients have either a BRAF variant or KRAS variant, and all the remaining patients, so around 56% of patients, do not have such a hotspot variant. So liquid biopsy or ddPCR could not give any clinical advantage or diagnostic advantage for those patients. And specifically for those patients, it is really critical to also establish untargeted ctDNA analysis, that those patients also could benefit from liquid biopsy analysis. And this is the next part of my presentation. So there I'm focusing on. So within the third paper of my PhD project, I established an approach called liquid biopsy, fragmentation, epigenetic signature, and copy number alteration analysis, or short LiveCNA. And this approach is based on the natural fragmentation of cell-free DNA. And by performing whole genome sequencing with around 6x coverage, I obtained information on fragment length and sequencing coverage. And from this information, I was then able, with bioinformatics analysis, to obtain further information on the global fragmentation within of the fDNA fragmentation in one patient, then the regional fragmentation, CRC-specific chromatin signatures, and somatic copy number alterations. And based on global and regional fragmentation, then in addition, I also trained a machine learning classifier or machine learning classifiers to assess whether sensitivity for ctDNA detection could even be improved. Overall, for the validation of LiveCNA or establishment of LiveCNA, probably 105 study participants were included, including 50 CRC patients. And of those patients, I was able to obtain 134 samples that were collected at a time point of the course of disease where the patients had clinically confirmed tumor evidence, and 61 healthy controls. And of those 61 healthy controls, 55 controls were included in the panel of normals. And rather than only establishing the method, I also wanted to assess how the clinical application or whether the method would be clinically valid. And therefore, I was able to obtain follow-up samples for 43 CRC patients. And for those patient samples were then, or those patients were then analyzed to assess whether live DNA could be used for residual disease detection or for treatment monitoring. First, I'd like to summarize the results I obtained for the global cfDNA fragmentation as an indicator for ctDNA. And the background or basis behind this is, in general, cfDNA fragmentation in healthy controls, as indicated here by the dark blue line, is expected to have a peak at around 167 base pairs. That is or can be associated to the length of DNA bound by one nucleosome plus linker DNA. And in cancer patients, it is expected that the peak, or that the fragmentation represents or has an enrichment in shorter cfDNA fragments. So as you can see here in this turquoise line, it would be expected to have a peak at a shorter fragment size. And this is actually what I also could confirm with my data. So compared to healthy controls in samples collected from patients with tumor evidence, I observed a significant enrichment of short fragments ranging from 90 to 150 base pairs. So just to say again, there was a significantly higher fraction of short fragments in CSC patients compared to healthy controls. Then when looking specifically at the results obtained for regional fragmentation, which might be another cfDNA feature that might serve as indicator for ctDNA, the background behind this would be that longer cfDNA fragments would be associated to closed chromatin, and shorter cfDNA fragments would be associated with active chromatin, so open chromatin. And when analyzing the data of patients included within this study, here is a summary plot showing only chromosome arms. But so on the x-axis are the different chromosome arms, and the y-axis represents the Z-score short to long ratio. And as you can see here in those on those chromosome arms marked with the arrows, there are multiple chromosome arms that show either a significant enrichment in short fragments or significant enrichment in long fragments compared to healthy controls. However, I'd like to mention, as in this study, only CSC patients were included, though those fragmentation profiles analyzed within this study are CSC specific and might look different for other tumor entities. The third cfDNA feature I analyzed with live DNA are CSC-specific open chromatin or CSC-specific chromatin signatures. And this is based on, regions that are expected to be specifically open in CSC, but not in healthy individuals. So in such regions, I would expect to have a constant coverage in healthy controls, as indicated here by the blue lines, and a drop in coverage in case of cancer patients. This could be also confirmed within my data. So I analyzed multiple region sets, and here on the left, you see in green the results obtained, the coverage profiles of healthy controls kind of for those region sets. And as you can see here at diagnosis, there were multiple patients where I observed those significant drops in sequencing coverage in those regions. In partial remission, actually, there are less patients with such significant coverage drops, but those coverage drops reoccur during progressive disease. However, also for samples collected from patients in remission, in some instances, there are still significant coverage drops compared to healthy controls that are still less significant compared to the true tumor samples. So overall, those results indicate that the detection of ctDNA based on active chromatin is applicable in samples with clinically evident tumor burden, specifically also at diagnosis. And as a fourth indicator for ctDNA with live DNA, somatic copy number alterations are analyzed. And here is actually the basic here, those dark blue dots show normal levels, or normal regions. And then it is expected that ctDNA represents actually with copy number losses or copy number gains as you can see here. And with this pipeline, I even analyzed focal amplifications that overlap with cosmic genes. And this is quite a complex plot, but those are in each line kind of all patients that were analyzed, and it was compared whether somatic copy number alterations were present in liquid biopsy only, in tumor tissue only, or in both liquid biopsy, so plasma samples, and tumor tissue. And as marked here with those arrows, there are already patients for which it was possible in stage 1 patients to identify overlapping somatic copy number alterations between plasma samples and tissue samples. So, and finally, rather than this normal kind of bioinformatics analysis, I also wanted to train a machine learning algorithm, and therefore I used the 134 samples collected from patients with tumor evidence, and a negative cohort consisting of the 55 samples from healthy controls that were included in the panel of normals, and 8 samples collected from patients that were already more than 6 weeks in remission, as those patients or samples are also expected to be collected from healthy individuals. And then, based on global fragmentation and regional fragmentation, machine learning classifiers were trained. And when looking at the results obtained for those, so there's one classifier in blue based on regional fragmentation data, one classifier based on global fragment size distribution, and the pink classifier indicates the results of a meta-learner combining both the global fragment size and regional fragmentation classifiers. And with all 3 classifiers, actually really promising results could be obtained, as the areas under the curve ranged from 0.93 to 0.95. And those classifiers reached sensitivities at 95% specificity from 63% to 73%, which is also really promising, as included our patient samples throughout all stages. Rather than only analyzing the basic performance of the different analysis, I also wanted to assess the performance of live CNA throughout the course of disease. And in this plot, the samples collected are categorized in colors depending on the time point of sample collection. So as you can see here in this red panel, those are samples collected at diagnosis, for example, and the dark color in each panel always indicates the presence of ctDNA or prediction of ctDNA, and the bright color are the samples for which no ctDNA was predicted on the specific cfDNA feature. And on the x-axis, so each bar represents one analyzed cfDNA feature. And I'd like to emphasize here that regional fragmentation was identified in this cohort to be the cfDNA feature with the highest sensitivity for ctDNA detection, as you can see here. And with the machine learning classifiers based on regional fragmentation and the meta-learner, a similar high sensitivity could be observed for samples collected at diagnosis. However, in case of the machine learning classifiers, when looking up here for the samples collected from patients in remission, specifically the first 6 weeks within remission, there were still high ctDNA predictions that disappeared more than 6 weeks post-treatment, kind of, to a range that might be also correlated with the clinic. So those false positive predictions, there are some possible explanations. It might be, for example, associated with epigenetic regulation or epigenetic mechanisms that occur in healing procedures, as those patients were recently or underwent recently surgery, or also with low molecular weight heparin that was previously associated already with shorter cfDNA fragments that was found in pregnant women, actually, that were tested with non-invasive prenatal testing. But this just as a side note. However, with live CNA, I didn't aim to analyze the presence of ctDNA or the performance of individual cfDNA features, but I wanted to assess whether, by combining multiple cfDNA features, whether the ctDNA prediction could even be improved, or the sensitivity could even be improved. And here again, I have plot sorted based on the disease stage where patient samples were collected at diagnosis, and each line represents one patient. And when highlighting those 3 patients marked here with the arrows and red boxes, you can see here, based on regional fragmentation data, that no ctDNA was predicted based on regional fragmentation analysis that I just mentioned in the previous slide was the cfDNA feature with highest sensitivity for ctDNA detection. However, by analyzing all the different cfDNA features, still when combining the analysis, as you can see here in this row, ctDNA was predicted in those patients or detected. And then I also have here with the purple background, the results obtained for machine learning classifiers. And when combining live CNA with the machine learning classifiers, so the bioinformatics analysis with the machine learning classifiers, the detection rate could even be increased. So overall, at diagnosis, it was possible to detect ctDNA in 81% of colorectal cancer patients with localized disease using live CNA alone, and in 93% of patients when combining live CNA with the machine learning classifiers. And in case of metastatic colorectal cancer, no difference was observed anymore, whether using only the bioinformatics analysis or live CNA in combination with the machine learning classifiers. And this, in combination with the results we obtained for patient samples collected in remission, indicate still that also live CNA without the machine learning classifiers is a really promising approach and really highly sensitive approach for ctDNA detection at diagnosis. So, and in addition to only focusing on ctDNA detection, I also wanted to analyze whether live CNA might be applicable for disease monitoring. And in this plot, you see in each line a different cfDNA feature that was analyzed to predict the presence of ctDNA. And those gray lines always indicate the cutoff above which presence of ctDNA was detected and below no ctDNA was detected. And when emphasizing those 2 time points here, this first time point, first arrow marks the time point from which no ctDNA was predicted anymore with live CNA. That was several months before the clinical finding of partial remission. And this arrow indicates the first time point from which in follow-up samples always ctDNA positive results were obtained, indicating again progressive disease several months before the clinical finding of progressive disease. So actually, live CNA also seem to work for disease monitoring. And overall, it was possible to predict response to treatment with live CNA in 77%. And when looking at CEA, so the reference biomarker, it was only possible in 25% of patients. And when looking at tumor progression, live CNA predicted tumor progression in all patients that were analyzed, and CEA only in 4 of the 5 patients that developed progressive disease at a certain stage. In addition, I performed one proof of principle experiment where I analyzed 6 additional healthy controls that were not included in the panel of normals to assess how specific the test might be. And I generated in silico dilutions ranging from 0.1 feature with the highest sensitivity for ctDNA detection, as already in 4 of 5 of those in silico dilutions, ctDNA could be detected with 0.5% tumor fraction. And when looking at the machine learning classifiers, the prediction of ctDNA or the performance seemed even to be lower than compared to the basic bioinformatic analysis. And in healthy controls, in 2 of 6 healthy controls, ctDNA positive results were obtained, or ctDNA was predicted, indicating low specificity of those machine learning classifiers. So in summary, live CNA was established for CSE, but by performing some adaptations to the bioinformatics pipeline, it is easily extendable to all solid tumors. With the bioinformatics analysis included in live CNA, by analyzing global fragmentation, regional fragmentation, CSE-specific chromatin signatures, and somatic copy number alterations, live CNA turned out to be highly sensitive and highly specific. And when looking at the machine learning classifiers, combining the results obtained for global and regional fragmentation, those classifiers are highly sensitive. However, for specificity, probably larger training and testing cohorts might be required, as this was quite small. So finally, I'd like just to make a short notice on the contribution of this PhD project. When looking again at the course of or ctDNA fraction throughout the course of disease of a patient, first establish ddPCR and live CNA, and I use distinct cutoffs for ctDNA quantification of ddPCR and follow-up samples, and I track changes in cfDNA features in follow-up samples using live CNA, and therefore with both methods, it is possible to perform treatment monitoring. And then by using distinct cutoffs for ctDNA detection, or in case of live CNA, also significance tests, both methods might be applicable for the detection of disease recurrence. Live CNA might have some issues with residual disease detection, but live CNA, as it is an untargeted approach, might even be applicable for the early detection of cancer. But how could liquid biopsy be implemented in clinical practice? And so far, probably the one point where it is already used and already arrived in clinical practice is the guidance of treatment decisions. And here, usually you have the diagnosis of a disease recurrence or also the early diagnosis of cancer. And usually targeted treatment selection is based on the analysis of tumor tissue. However, in some instances, it might just not be feasible to obtain a tissue biopsy, or the long turnaround time until you have an appointment, or low quantity or quality of material does just limit the performance and you're not able to obtain results. And in those cases, actionable variants could actually be detected based on the analysis of circulating tumor DNA. And since here only distinct hotspot variants are used for targeting treatment, the ddPCR might be the method of choice. Then another interesting approach that I was working on actually within this PhD project is the detection of residual disease or recurrence. And in this case, you have a patient that undergoes surgery. A blood sample would be collected following surgery, and based on the presence of ctDNA or absence of ctDNA, the decision for adjuvant chemotherapy might be guided. And this can be done with ddPCR, as I showed within the second paper of my PhD project. And it might be done if it's longer or more than 6 weeks post-surgery, also with live CNA. And then a third interesting approach for how liquid biopsy could enhance or could be used in clinical practice is treatment monitoring, as by decreasing ctDNA levels, response to treatment could be predicted. By stable ctDNA levels, stable disease could be predicted, and by increasing ctDNA levels, progressive disease could be detected. So actually by tracking changes in ctDNA, treatment monitoring might be applicable. And this can be done with both methods I established and validated throughout my PhD. With this, I'd like to thank again my supervisor for giving me the opportunity to perform my PhD project at Medical Genetics Center. It was really great time. And also my complete TAC for their support throughout the process, for guiding me. And also all the other people that were involved in this project and supported me throughout the time. And probably there are way more people that weren't involved in this project that also kind of mentally supported me. So thanks a lot, and I'd be happy to answer your questions if you have any.